Staphylococcus aureus class 2. Staphylococcus aureus can ferment a number of sugars but produces only acid, no gas. Usually, this sugar fermentation is of no diagnostic value, or we cannot use this sugar fermentation property for the diagnosis of the bacteria, but except for mannitol, because which is usually fermented by Staphylococcus aureus, not by other species. So, to specially determine Staphylococcus aureus, this mannitol fermentation is a criteria. This bacteria is catalase test positive. They can hydrolyze urea. They can reduce nitrate into nitrite. They have gelatin liquefaction liquefaction property, and they are MR test and VP test positive. They have lipolytic ability. Since they have this lipolytic ability, when they grown on media containing egg yolk, they produces dense opacity, or they convert this media into a dense opaque form. Okay, they are able to produce phosphatase enzyme. So this phosphatase enzyme production can be determined by culturing this bacteria on nutrient agar medium containing phenolphthalein diphosphate. When such a culture is exposed to ammonia vapor, this colony is converted into a bright pink color due to the presence of free phenolphthalein. This is useful for the screening procedure, especially for differentiating Staphylococcus aureus bacteria from Staphylococcus epidermidis in a mixed culture. Another important biochemical property of Staphylococcus aureus is that they are able to produce thermostable or heat tolerant nuclease enzyme. We know that the function of nuclease enzyme is degradation of nucleic acid. Among the non-sporing bacteria, Staphylococci are more resistant. When dried on threads, they remain viable for 3 to 6 months, and dried pus, they can retain their viability for 2 to 3 months, and they may withstand 60 degrees Celsius for 30 minutes. Their thermal death point is 62 degrees Celsius for 30 minutes, and they grow well in media supplemented with the 10% to 15% NaCl, and this feature are of significance in food preservation. For example, for preserving food like pickles and all, we used to add high concentration of salt, but this bacteria can cause contamination of such food since they can withstand 10 to 15% of NaCl concentration, and they resist 1% phenol for 15 minutes. Mercuric perchloride, especially 1% solution, that kills them in 10 minutes, and anilyl dyes, crystal violet, brilliant green, this type of stain are also inhibitory or lethal to the bacteria. Fatty acids, especially present on our skin surface, these fatty acids are highly inhibitory to this bacteria, especially unsaturated fatty acid. They can resistant lysosomal action, but they are sensitive to lysostaffin. Okay, this lysostaffin means it is a mixture of enzyme produced by a particular strain of Staph epidermidis. So this lysostaffin can inhibit Staphylococcus aureus. And these bacteria are uniformly sensitive to penicillin. Though occasional strains from the pre-antibiotic era have been found retrospectively to be capable of producing penicillinase enzyme, and they are able to resist penicillin antibiotic. Let us study the mechanism used by Staphylococcus aureus to resist penicillin antibiotic. The first mechanism is the production of beta-lactamase or penicillinase enzyme. We know that the penicillin antibiotic, structurally it contains a beta-lactam ring that make it functional. So this penicillinase or beta-lactamase enzyme specifically cleave this beta lactam ring and make penicillin drug inactive. Staphylococci are able to produce mainly 4 types of penicillin A, B, C and D. But hospital strains, they predominantly produce type A penicillin. The second mechanism used by bacteria is they create changes in bacterial surface receptor. The staphylococcal bacterial cell on their surface, they contain specific receptor. Actually, the penicillin drug go and bind to the specific receptor and then they act over this bacteria. But this Staphylococcus aureus, in order to resist penicillin antibiotic, they roughly changes the properties or the structure of this surface receptor. Then the penicillin antibiotic cannot bind over it and they cannot execute their function. Third mechanism is development of tolerance to penicillin. The unwanted and continuous use of an antibiotic like penicillin create tolerance ability in the bacteria, but by which the bacterium is only inhibited but not killed. Antibiotic resistant property not only to penicillin, sometimes it extends to cover some other beta lactamase antibiotic also. For example, methicillin and cloxacillins. So some of these strains, they may show resistant to other antibiotics and heavy metals. They can cause great problem or severe outbreak in hospital infection. These strains are particularly known as EMRSA. EMRSA means epidemic methicillin resistant Staphylococcus aureus. Actually, the resistance of Staphylococcus aureus is plasmid borne. That means the Staphylococcus aureus in their cell carry a plasmid. This plasmid allow the or plasmid gives the ability to resist this antibiotic to the bacterium. So this property is provided by plasmid, so it is known as plasmid bond. So this plasmid bond resistance is not only shown to penicillin, but it can also show resistance to some other antibiotic like erythromycin, tetracycline, aminoglycosides and almost all against all clinically useful antibiotic except the antibiotic vancomycin. Pathogenicity and virulence. Generally, Staphylococcus produces two types of diseases, either infection and intoxication. Okay, in case of infection, the cocci that directly causes the disease, the bacterium itself causes the disease. The cocci enter our body through damaged skin, mucosal or tissue sites. Then they adhere firmly or they attach firmly to our tissues. There they colonize us and they causes or they overcome the host defense mechanism and they started multiplying on the site, and there they causes tissue damage and produces localized suppurative lesion or generally infection. In case of intoxication, the disease is caused by bacterial toxin, not by the bacteria. Either this toxin is produced by the bacterium whenever the bacterium as such entered into our body, and otherwise the toxin can enter into our body when we are having food contaminated with this bacterial toxin. Virulence factors, that means these factors helps the bacterium to develop the disease. So it mainly include 5 categories of virulence factor. First one, cell associated polymers. Next, cell surface protein, extracellular enzyme, protein receptors and toxins. Let us study one by one. First, cell associated polymers. There are mainly 3 cell associated polymers that allow the bacterium or that helps the bacterium to produce disease. First one is cell wall polysaccharide. The major cell wall polysaccharide of Staphylococci is peptidoglycan. The major function of peptidoglycan is that it provides a structural integrity and rigidity to the bacterial cell. Second is teichoic acid. Teichoic acid is an antigenic component present in the cell wall of the bacterium. By using the teichoic acid, bacteria attaches firmly to the host cell surface. Okay. Next is capsular polysaccharide. This capsular polysaccharide, we know that capsule usually surrounds the cell wall, and this capsular polysaccharide protect the bacterium from the phagocytosis and opsonization. These two are the mechanism used by our body or host cells to resist the bacterium. The second category of virulence factors is cell surface protein. There are mainly two types of cell surface protein, protein A and clumping factor. Protein A is chemotactic. Chemotactic means they attract all the Staphylococcus aureus strains and allow their colonization in an area. So that property is known as chemotactic, and they are anti-phagocytic. That means they resist phagocytic property of the host, and anti-complementary. We also use complement protein to resist or to kill the bacterial cell. So this protein A gives the bacterium anti-complementary effect. And they also causes platelet damage, hypersensitive reaction in our body. Next is clumping factor. Clumping factor are also known as bound coagulase. That means they are coagulase enzyme remain attached to the staphylococcus cells and not released. So this, by using this bound coagulase, we are diagnosing this staphylococcus bacteria mainly by slide agglutination test. So in slide agglutination test, human plasma is mixed with the saline suspension of Staphylococcus aureus, and if it is saline Staphylococcus aureus, definitely it will produce bound coagulase and that clump the human plasma. So it gives slide coagulase test positive. The next category of virulence factor used by the bacterium is extracellular enzyme. The bacteria produces different types of enzyme that facilitate the bacteria in order to cause the disease. The first group of enzyme produced by the bacteria is coagulase enzyme. Coagulase enzyme, their property is that there are mainly two types of coagulase enzyme produced by bacterium. First one is bound coagulase, second one is free coagulase. Bound coagulase we have already discussed, it is otherwise known as clumping factor. So here we are discussing about the free coagulase. So this coagulase enzyme are capable of clotting human and rabbit plasma. In order to become functional, this coagulase enzyme requires CRF, coagulase reacting factor, which is present in plasma. Okay, so this coagulase enzyme produced by Staphylococcus aureus combined with the coagulase reacting factor CRF, and they are together able to convert fibrinogen into fibrin. We know that this fibrin helps to produce clot. Different category of coagulase enzymes were produced, but most human strains of Staphylococcus aureus produces coagulase type A. This free coagulase and clumping factors are different in the sense this free coagulase requires CRF, clumping factor doesn't require CRF, and this free coagulase is secreted into the medium. Bound coagulase always remain attached to the bacterial cells, not freely released into the medium. Next, lipase. Staphylococci produces a number of lipid hydrolase or lipase enzyme which helps them to infect our skin and subcutaneous tissues. Hyaluronidase enzyme, they breaks down connective tissue and they helps to spread the infection or helps to spread the bacteria in our body. Next is Phyllo Kinase. This enzyme is also known as fibrinolysin. If any clot is produced by our body, the bacteria become trapped there. But this bacteria produces fibrinolysin or Staphylokinase enzyme, and they breaks down this fibrin clot, and thereby they can freely move in our body. and we know that fatty acids are inhibitory to this bacteria, so they produces fatty acid modifying enzyme. Next is protease enzyme. They causes the initiation and spread of the infection. Nuclease enzyme, especially, they produces heat stable nucleases. So then after enzymes, the another category of virulence factors is protein receptor. The category of virulence factors is protein receptor that facilitates Staphylococcus attachment. Staphylococci bacteria, they are able to produce different types of toxin, generally classified into cytolytic toxin, Enduro Toxin, toxic shock syndrome toxin, and exfoliative toxin. So the first toxin we have to study is alpha hemolysin. Alpha hemolysin is the most important category of hemolysin. Generally, hemolysin means they are able to lyse or degrade RBC. This alpha hemolysin is having a special property, that means they are inactivated at 70 degrees Celsius but reactivated at 100 degrees Celsius. Normally what happens at higher degree temperature, denaturation occurs, but here at lower temperature they are inactivated, but higher temperature causes the reactivation of the toxin. How means at 70 degrees Celsius, the toxin is bound and inhibited by an heat labile or heat sensitive inhibitor. When the temperature is increased to 100 degrees Celsius, this inhibitor is denatured and the toxin become free and activated. This alpha hemolysin is also having some other property, leukocidal. Leukocidal means they kill leukocytes. Cytotoxic, they are toxic to normal cells. Dermonecrotic, they causes necrosis of skin cells. Neurotoxic, they are toxic to nerve cells, and they are lethal. Lethal means causes the death of any type of cells, and they are toxic to macrophages, lysosomes, muscle tissues, renal cortex, etc. This is the typical image showing alpha hemolysis of staphylococci. Or when you observe, you can see that green colored colonies or green colored zone you can see. Why? This is because alpha hemolysis is a partial hemolysis. That means they usually hemolysis is tested by growing this bacteria in blood agar medium. So whenever we are cultivating this bacteria in a blood agar medium, if they are showing alpha hemolytic property, you can see the colonies and surrounding zone in a green color. It is due to the partial degradation of RBC. So alpha hemolysis in which the zone is appeared in green color due to the partial degradation of RBC. Type of hemolysin to be studied is beta hemolysin. Beta hemolysin is able to degrade this RBC completely. Okay, so that whenever this bacteria carry out beta hemolysis in blood agar medium, the zone appear very clear and transparent, or pearl yellow colored halo is appeared around the bacteria. This beta hemolysin is evident from this image, and this beta hemolysin is also known as sphingomyelinase, and especially this beta hemolysin is hemolytic to the sheep RBC, not for human and rabbit. This beta hemolysis or the beta hemolysin, they show hot cold phenomenon. That means they start degrading RBC or they start hemolysis at 37 degrees Celsius, but this zone of degradation can be seen only after chilling. This phenomenon is known as hot cold phenomenon of beta hemolysis. The type of hemolysin is gamma hemolysin. Structurally, this gamma hemolysin is a bicomponent and it is composed of two separate proteins. Usually, gamma hemolysis means no hemolysis. That means they are unable to degrade RBC. That when we are cultivating this bacterium in a blood agar medium, and if they are showing gamma hemolysis, no zone can be seen around the bacterial colonies. Next is delta hemolysin. This delta hemolysin is having a detergent effect on the surfaces of cells like macrophages, erythrocytes, leukocytes, and platelets. And we already studied that cytotoxic category, cytolytic toxin category include 4 hemolysin and 1 leukocidin. So next one is leukocidin. Leukocidin is also known as Panton-Valentine toxin. It is also a bicomponent toxin. It damages the membrane of cells, and this leukocidin and gamma hemolysin, they are together known as synergohymenotrophic toxin. Next category of toxin is Enduro Toxin. Enduro Toxin means they affect intestinal cells, specially known as enteric cells, so that they are named as Enduro Toxin. Enduro Toxin causes staphylococcal food poisoning. The major symptoms include nausea. Nausea means tendency to vomit, and then vomiting, diarrhea. The symptom usually appear 2 to 6 after consuming the contaminated food. The contaminated food may contain bacterial toxin. So this enterotoxin is heat stable, and especially meat, fish, milk or milk products cooked or left at room temperature may be contaminated with this Staphylococcus enterotoxin. So this accumulated toxin causes infection to us. The source of infection is usually a carrier. Carrier means the person who carry this bacteria in their body, and if the carrier is a food handler, it increases the risk. The disease is not fatal, it is self-limited. There are mainly 8 antigenic types of enterotoxin. It is given here, but among which type A is most common. Usually, in order to find out this toxin presence in our food, we can use latex agglutination or ELISA test. Next is TSST or toxic shock syndrome toxin. It is a potentially fatal toxin. It causes multi-system disease, and the symptoms mainly include fever, hypotension. Hypotension means low BP. Myalgia. Myalgia means general weakness of the body. Vomiting. Mucosal hyperemia. That means blood vessels to the mucosal tissues are filled with a large amount of blood. And erythematous rashes. That means red colored rashes appears on our skin. The disease caused by this toxin is known as TSS, toxic shock syndrome. TSS was first identified in 1978, especially among children and adolescents. And a special type of TSS is known as tampon-related TSS, common in women. Last category of toxin is exfoliative toxin. Toxin is also known as epidermolytic toxin, ET toxin, exfoliatin, etc. They causes a disease known as staphylococcal scaled skin syndrome, SSSS. It is an exfoliative skin disease. That means the outer layer of our skin is separated from the underlying layer. The severe form of SSS is known as Ritter's disease. This Ritter's disease is commonly seen in children or infants.